you just have a touch or have a line or underline that. Okay, so this is a question on inhibition of enzyme. So let me about the enzyme, let me tell you one thing. Now from the last year onwards, we have 7 classes. We were divided into 7 classes. Enzymes based on its functions, based on its type of reaction, it helps. Earlier, you were studying about 6 classes. Oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. Now we have one more. It's called the translocase. So, 7 classes. Out of that, the 7th class is on isomerase. Then last one, lyase, isomerase, ligase, and translocase. So, enzyme 1 means oxidoreductase, enzyme class 2 is transferase, enzyme class 3 is hydrolase, enzyme class 4 is lyase, enzyme class 5 is isomerase, enzyme class 6 is ligase, and 7th one, I put it in red color, translocase. You might be familiar with the word carnitine, acyl, carnitine, translocase, and all. So that comes under that. Proton pump, that all comes under translocases. So I want to inform about that. And then we'll come to the question about enzyme inhibition. Mainly, we have the 3 types of inhibitions we know. We have different types in that. The first 3, about the commutative inhibition. What do you mean by commutative inhibition? Here, the inhibitor is a structural analog of substrate, and it will be binding to a site called as active site. So there is a competition between the inhibitor and the substrate to bind at the active site. So they are structural analogs, structurally similar. It binds only to enzyme, and then this reaction is mostly reversible. Commutative inhibition is reversible reaction. It becomes reversible if you increase the concentration of substrate. Here, in this reaction, see, there's no change in the structure of the enzyme. So there is no change in its maximum velocity that it can attain. That's called as Vmax. That remains same. But Km value, what is Km value? Concentration of the substrate to attain half the maximal velocity is called as Km value. That is getting increased. And then one clinical importance is most of the actions of pharmacological action of drugs is by commutative inhibition. So from the question now we are able to understand what's the answer for it. Anyway, I'm just trying to compare the other things also. Commutative, we have non-commutative now. Non-commutative, see, in commutative they were structural analogs. Here they are not structural analogs, so there is no competition. And it binds to a site other than the active site. That's one difference. And another difference is in non-commutative inhibition, the inhibitor can bind either to enzyme or to enzyme substrate complex. And it is reversible or irreversible. Generally, it is irreversible. And here, there's a change in the shape of the enzyme, because of which Vmax is decreased. Km value remains same. And here in non-commutative inhibition, the most important thing is most of the toxic compound, poisons, acts by non-commutative inhibition. One example is our cyanide that inhibits cytochrome oxidase. The third one is about the uncommutative inhibition. Here, as you know, it's not a structural analog, there's no combination, and it again binds at another site. But here, one major difference is it can bind only to enzyme substrate complex. I'm sorry, I cannot change this one. Yeah, it can bind either to, only to enzyme substrate complex, and then this is irreversible, no doubt in that. Both are decreased. Vmax and Km values are decreased. So these are the major differences between the commutative, non-commutative, and non-commutative. So if a question can come on commutative, then naturally the question can be on non-commutative or uncommutative. Okay, so I hope now you can able to understand the answer. It is easy now, which is not true. And then other than these 3, we have one more. We have another one more is there. We call it as suicide inhibition. That also you should remember. Okay. Now the second question. Sugar of milk. Here, again, the question is on chemistry of carbohydrate. Sugar of milk, as you know, it is lactose, which is made up of glucose and galactose. Now, what are the other questions that can come in this part? What about the sugar of the table sugar? A carbohydrate percent the table sugar, it is nothing but sucrose. And then what's the sugar in milk? That we know just now we said lactose. In rice, what's the sugar? Starch is a polysaccharide. What about the sugar in sprouted seeds? It is maltose. In green leafy vegetables, the carbohydrate percent is mainly dietary fiber. We call it that because it's cellulose and all, which are indigestible form of carbohydrate. So generally, any indigestible carbohydrate, we use the word dietary fiber. Example is cellulose, hemicellulose, lignin. So green leaves contain that. So remember it. And another, the major carbohydrate that are present in the extracellular matrix. Extracellular matrix is mucopolysaccharide or glycosaminoglycans. So have a read on that. You can have a glance on that. Major types of extracellular glycoprotein, protein, we call it as proteoglycan because carbohydrate is more, protein level is around only 5%. Proteoglycans and mainly mucopolysaccharide. And the names are chondroitin sulfate, mainly present in the cartilage, hyaluronic acid in the synovial fluid, keratin sulfate in the cornea, then heparin sulfate is there in the membrane, our extracellular glomerular membrane, again extracellular matrix only, heparin, an anticoagulant. So these are some examples for heteropolysaccharide that are mainly component of extracellular matrix. Okay, so third question. GLUT2, we call it as glucose transporter 2, is seen in all the following tissues except. First of all, what is GLUT? It's a transporter. Means what? It's a transmembrane protein. What do you mean by transmembrane protein? A protein what we can see from the both sides of the membrane. A transmembrane protein that helps in the transport of glucose. It's called a glucose transporter. Okay, then transport of glucose occurs mainly in there are two types of transporters, we can say like that. One is sodium dependent transporter, where we have an active transport. That means ATP dependent. It's mainly secondary active transport. First in the intestine and kidney, and is sodium dependent. And we have another one, facilitated transport. They are the GLUT1 to GLUT7 we have. And that is the one that's present in almost all tissues. Okay, so I'm just trying to say the difference between sodium dependent and sodium independent. Sodium independent means, as I said, facilitated diffusion. Sodium dependent is secondary active transport, but sodium independent is a passive transport. Here, sodium dependent is symport. That means what? Sodium and glucose together moves in one direction. It's transported in one direction. We require ATP, and that is linked to our sodium potassium pump in another side. That's why the word secondary active transport. Okay, then of course from the word itself, it requires the sodium. And here the transportation can be against the concentration gradient also. But in sodium independent, it's passive. Okay, major ones, the sodium dependent are S-GLUT1 and S-GLUT2. S-GLUT1, S-GLUT2. Sodium dependent glucose transporter 1 and sodium dependent glucose transporter 2. Sodium dependent glucose transporter 1 is there in the intestine for the absorption of glucose after the digestion of carbohydrate. And S-GLUT2 or sodium dependent glucose transporter 2 is made in the renal tubules that help in the reabsorption of glucose that has been filtered by the glomerulus. Okay, and whenever there's some defect in the functioning of S-GLUT2, that leads to the word called as renal glycosuria. The word that we use, if it's specific glucose, then we use the word renal glucosuria. Okay, so as GLUT2 different. defect leads to renal glucosuria. Please remember that. What is renal glucosuria? That means even if the blood sugar level are in the normal range, then even in that condition also glucose is present in the urine because the reabsorption is not proper. That happens mainly because of the defect of SGLUT2 and also in Fanconi syndrome, another one. Okay, sodium independent transport is dependent was active transport. Here it is passive transport. And here only it's a unipod. Energy is not required for this process. And it doesn't require sodium also. And transportation is along concentration gradient. We have the glucose transporter maybe from 1 to 7. I'll just show that also. Transport of glucose from blood to tissues is by this GLUT1 to this one. So GLUT1 is mainly present in the RBC, brain, kidney, retina, and placenta. GLUT2 you can see in the liver, beta cells of pancreas, and small intestine. So that was the question. It was the exception. The question was except. So these three are the one which contain GLUT2. GLUT3 in brain and neurons. And that another question that can be related to the glucose transporter can be, which is insulin dependent. GLUT4, which is present in the skeletal system, heart, and adipose tissue, is insulin dependent. That means in type 1 or type 2 dependent diabetes mellitus, what happens? The GLUT4 will not be active. It won't be able to transport the glucose into the skeletal system, heart, or the adipose tissue. That's the point. So this is insulin dependent. That's one important things to be noted. GLUT4 is there, the small intestine also. That's the one which helps in the transport of fructose or absorption of fructose. Is present in the testis also. GLUT7 for liver, present in the liver, endoplasmic reticulum. So I hope that this third question we understood. Again, the question was an exception, so you can understand the question now. Now I hope you will be able to answer the question now. Fourth question, which of the following is the rate limiting enzyme in glycolytic pathway? We have 4 options. Of course, you know, in this, only D comes under that in the glycolysis, phosphofructokinase. So I'll just, now it's a question from the carbohydrate metabolism. We'll just have a quick look on some points. First about glycolysis. That means, glycolysis means what? Breakdown of glucose into pyruvate and then lactate in the case of anaerobic conditions. We have 2 types of glycolysis, aerobic and anaerobic. In aerobic, we get 7 ATP. In anaerobic, we get 2 ATPs. And the products are, in aerobic condition, the product is pyruvate. In anaerobic condition, the product is lactate. 3 major points about glycolysis. I'll say the 1, 2, 3 of glycolysis. There is one shunt pathway bypass in that glycolysis that happens only in the RBC for the production of 2,3-DPG. And second point is that there are 2 substrate level phosphorylation. That means in glycolysis, we have 2 steps where ATP is produced straight away. 2 substrate level phosphorylation and 3 rate limiting enzymes. Rhymically, we say HK, PFK, PK. That means hexokinase, phosphofructokinase, and pyruvate kinase. And that the PFK, phosphofructokinase, is the most important one. That's what the questions asked also. Phosphofructokinase. So 1, 2, 3. 1 shunt pathway, 2 substrate phosphorylation, and 3 rate limiting enzymes. Understand that glycolysis, and one more question here. One more question that can be asked. The sole source of energy for RBC is glycolysis. That's the only source of energy for any RBC is glycolysis. Please note that. That's also an important point to be noted. Okay. Now, in the same way, the question can be asked about other pathways also. A pathway called as gluconeogenesis. It is about the synthesis of glucose whenever we are in need of glucose. And we have 4 enzymes we have. There comes the glucose 6-phosphatase, fructose 1,6-bisphosphatase, phosphoenol pyruvate carboxykinase, and pyruvate carboxylase. That 2 enzymes given A and B comes under for gluconeogenesis, please note. So, and glucose 6-phosphatase, pyruvate carboxylase. Other than that, I said two more. They are fructose 1,6-bisphosphatase and phosphoenol pyruvate carboxykinase. That are the four major enzymes of gluconeogenesis. And the question can be on glycogen metabolism. Glycogen metabolism, the two major things are there in glycogen metabolism. One is glycogenesis, synthesis of glycogen. There the enzyme is glycogen synthase, and in glycogen breakdown or glycogenolysis, the main enzyme is glycogen phosphorylase. And we have one more pathway regarding the glucose major pathway called as HMB pathway or hexose monophosphate pathway or pentose phosphate pathway. There, the rate limiting enzyme is glucose 6-phosphate dehydrogenase. So if they can ask you one question, the similar question can be the rate limiting enzyme of other pathways also. That's why we had a just quick look on these questions. So I hope now you understood the answer for the fourth question. Answer is D, phosphofructokinase. Okay, one more question that can be asked related to glycolysis is about in the reality, in the clinical conditions, whenever we take the blood sample for glucose estimation, we will always take the blood sample in fluoride bottles. Why fluoride bottles? Because fluoride can inhibit the glycolysis in RBC by inhibiting the enzyme enolase. So there's a chance of reducing glucose on keeping will be stopped, will be inhibited by this process. Okay, that means what? To maintain the blood sugar level, to stabilize the blood sugar level in the test tube, we will add the fluoride that inhibit the enzyme enolase, a second last step of glycolysis. Please note that also. That's also another question. Okay, going for the fifth question. Von Gierke's disease or type 1 glycogen storage disease is due to the deficiency of. This is about glycogen storage disease, glycogen metabolism. See, glycogen usually breaks down and we get the glucose. Glycogen is the storage form of glucose, you know that. Glycogen is mainly the storage form of glucose and is stored mainly liver and muscle, that you know. So it's a homopolysaccharide made up of glucose. It's a branched homopolysaccharide, more better word, branched homopolysaccharide, storage form of glucose and mainly stores in liver and muscle. And then liver glycogen we use to maintain blood sugar level. But the glycogen that is stored in muscle is only for the energy for the muscle alone. It is not for that muscle glycogen cannot be used to maintain blood sugar level. Meaning, when you catabolize the glycogen, you get glucose. That glucose will not come out of muscles. Why? Or other words, if it comes out of the muscle to the blood, then thereby it can maintain the blood sugar level. Okay, and then why it cannot? Because of the absence of one enzyme in the muscle, it is glucose 6-phosphatase. That's present only in liver, okay? It's not there in the muscle. So the glucose 6-phosphate cannot be converted to glucose. Then only it can come out of if it becomes glucose, then only it can come out of the tissues. So in the case of muscle glycogen breakdown process, glucose 6-phosphate is not converted to glucose because of the absence of enzyme glucose 6-phosphatase. So it cannot be used to maintain blood sugar level. Okay. You know, I said about there are two types of pathways in glycogen metabolism. One is the anabolism, it's called as glycogenesis, and the rate limiting enzyme is glycogen synthase. And in the case of glycogen genolysis or glycogen breakdown, the main enzyme is glycogen phosphorylase. Okay, rate limiting enzyme. Now, whenever glycogen is not degraded as per our need in our body, it accumulates in the muscle or the liver, and that's called as glycogen storage disease. Okay, the major types of glycogen. So here I'll say, okay, von Gierke disease, we'll just go to the answer later. So these are the major glycogen storage diseases. Type 1, the enzyme deficiency glucose 6-phosphatase. Type 2 is Pompe disease and the lysosomal maltase. Type 3 is Cory's disease and D branching enzyme. Type 4 is Anderson disease branching enzyme. You can just study this way: CDAB, Anderson, A stand A letter, branching enzyme. Branching enzyme for glycogen synthesis, debranching for glycogen breakdown. You know, understood that. McArdle disease, the muscle phosphorylase. Hers disease, liver phosphorylase. And type 7 is called as Tory's disease, muscle phosphofactor kinase. 8 and 9 is don't have any names as such. 8, glycogen storage disease 8, is due to the deficiency of liver phosphorylase kinase. And other one, type 9, is about the muscle phosphorylase kinase. So now I hope you can say the answer now. Our type 1 von Gierke's disease is because of the deficiency of glucose 6-phosphatase. Going for next question regarding HMP pathway. All of the following are true except, please underline that, except. Okay. What about HMP pathway? So far we said about glycolysis, gluconeogenesis, glycogen metabolisms. And they mainly somewhat always related to energy, like glucose storage or glycogen breakdown, glucose breakdown, thereby energy. But HMP pathway, that's the most important point. HMP pathway is not for energy. No ATP is produced, not for energy. That's the point to be noted. Then for what? It's mainly for NADPH. We call it as reduced coenzyme or reducing, generally we say reducing equivalent of the body. Energy currency of the body is ATP. The same way, the reducing currency of the body, we can call it as NADPH. NADPH and pentose phosphate are the two products of HMP pathway, or that's the use of HMP pathway. HMP pathway is just for the synthesis of NADPH and for pentose phosphate. It's not for ATP synthesis. That's what you should know. Takes place in cytoplasm, and in the there are two phases for HMP pathway: oxidative phase and non-oxidative phase. In non-oxidative phase, just what happens is just pentose phosphate will be converted to another type of pentose phosphate pathway. Just in the conversions is only taking place, and later we get a fructose 6-phosphate also. So here the answer except is C, non-oxidative phase generates pyruvate. That's the wrong answer, wrong option given here. We'll just have a quick look on it. HMP pathway, it takes place in cytoplasm, mainly for NADPH and pentose. Here, as I told you earlier, the rate-limiting enzyme is glucose 6-phosphate dehydrogenase. G6PD is called like, we call like that, G6PD, glucose 6-phosphate dehydrogenase. It has two phases, as I said, oxidative and non-oxidative phase. Oxidative phase produce NADPH and pentoses. Non-oxidative phase just in the conversion of pentose phosphates. No ATP, please note, is no, for this pathway is not for energy. Okay, so this what another one about the significance. One important part is the use of HMP pathway. We said earlier that we can say two types of significance: biochemical and clinical. And biochemically, we have mainly NADPH and pentose phosphate. This NADPH is used for any reducing steps. Our understand one most basic question, long back to the PG question even. All the catabolism that takes place in the body is oxidation, and anabolism is reduction. Okay, and most of the synthesis pathways requires NADPH because it's a reducing currency, lipid biosynthesis. Then almost all lipids, in the sense fatty acid, triglycerides, phospholipid, steroids, cholesterol, in all we need NADPH. Okay, and then free radical scavenging. Free radicals can cause the islands, can become opaque of that, opaqueness. And now when it prevents the free radical action or that helps in free radical scavenging, thereby NADPH is helping in the islands transparency to maintain the islands transparency and help in the conversion of methemoglobin to hemoglobin. As you know, methemoglobin is the hemoglobin with iron in ferrous state. It cannot carry oxygen. For this conversion, we need NADPH. And the same way, by scavenging the free radicals or by inactivating the free radicals, it helps to maintain the erythrocyte membrane integrity. So indirectly, HMA pathway helps in all these processes. That's what I want to tell you. The question can be in that way also. And this NADPH helps in detoxification process in our body. The major enzyme for detoxification is cytochrome P450 that happens in liver. For that, we need NADPH. I repeat that enzyme name, cytochrome P450. And then bactericidal action. I'll just come to that word again with another question. NADPH helps the macrophages to kill the bacteria. Okay, clinically, if the G6PD is deficient, clinically, in a patient, if a G6PD is absent, then the one thing mainly happens is drug-induced hemolysis. So enzyme deficiency that leads to drug-induced hemolysis is G6PD deficiency. And as I said earlier, it helps in the conversion of methemoglobin to hemoglobin. So G6PD deficiency leads to accumulation of methemoglobin. So it's another word, methemoglobinemia. So just try to cover most important points in HMB pathway. As I said earlier, see, one of the main function of NADPH is that it helps the tissue macrophages to phagocyte and then to kill the bacteria. How we do that? This, through the HMB pathway, NADPH is produced. NADPH, with the help of enzyme NADPH oxidase, converts oxygen atom into superoxide. That superoxide is highly reactive. It can kill the bacteria. Or otherwise, superoxide converts to hydrogen peroxide, then with the help of chloride, hypochlorous acid. That can also kill the bacteria. So in two ways, NADPH help in a process we can call it as oxygen-dependent killing. So seventh question, oxygen-dependent killing is done through which enzyme? The enzyme name given here, mainly NADPH oxidase enzyme. So HMA pathway, now we hope you can understand the function of HMA pathway, produce NADPH that has a bactericidal action or that is involved in oxygen-dependent killing. That's another question that has been asked in the exams earlier. Hope this process is clear for you. Now about another question, immunoglobulin that mediates primary immune response. Major immunoglobulins name are given as options. We'll just go through one by one each. Which is the natural antibody? Is IgM. Natural antibody in the sense, very simple words. A patient, the simplest one, a patient with blood group A, he will be having A antigen and B antibody in his body by birth itself. So that B antibody has come naturally. That natural antibody is IgM. IgM is a natural antibody, and that's the first type of antibody that is produced whenever we get exposed to any antigen. Why? I should say in this way, what's the important significance of it? IgM is a pentamer. So it will be having 10 sites to bind to the antibodies. So the antigens. 10 antigen binding sites. So because of it, that's more effective in the phagocytic reactions. IgM, natural antibody. It's a primary antibody. It's a pentamer. So it has 10 antigen binding site. And these 5 units are linked by JH chain. And one more, 10 antigen binding sites it has. Okay. Then another one is IgG. IgG that's a predominant one. That's the most abundant antibody present in our body. And that's a secondary. So second, after the IgM, this is the antibody that is produced. And then it's a monomer. IgG is a monomer. And so since the monomer, there's no need of J chain here. And this is the only one that crosses the placenta. So mother to fetus. So whenever I teach a student, I'll say, in Hindi, what do you call for your mother and father with respect? Madaji, Pidaji. I'll say that G stands for IgG. That's an easy way to remember the things. This is the only immunoglobulin that is transferred from the mother to fetus. Okay, next question is about, next one. Next antibody is IgA. IgA is a dimer that's present in almost all secretions. So we call it a secretory antibody. IgA is secretory antibody. It has a secretory component because of which it cannot be digested. It cannot be digested by the digestive enzymes. It's a dimer, again linked by through the J chain, and it is present in the colostrum also. And another major antibodies, as you know, is IgE. IgE is major is the least abundant antibody. It's cytophilic antibody, and then it's a monomer. This is the most important point. It mediates allergic reactions, or other hypersensitive reactions. Mediates hypersensitive reactions. So this is another important point about IgE. You should remember. So these are the major questions that can come on this one. Which is the major natural antibody? Which is the major primary antibody? Which is the abundant antibody? Which is secretory antibody? Which antibodies crosses the placenta? Which antibody is present in the colostrum? Which antibody present in all secretions? And which antibody mediate the allergic reaction, type 1 hypersensitive reactions is IgE. So from these things, I hope you understood the answer for the 8th question. Immunoglobulin that mediates the primary immune response is IgE, is the natural antibody. 9th question. The protein digesting enzyme present in the stomach is about the digestion part, digestion of protein. Think from here. This is one question that can be asked. All proteolytic enzyme is secreted in the inactive forms because it shouldn't digest our own cells. It will be in the inactive form. The inactive form of an enzyme, we use the word zymogens. And there are two types of enzymes that can break the peptide ligages. It can be endopeptidases, exopeptidases. Endo means that breaks inside a long chain. Here and there, randomly we can call it as endo. Exopeptidases means it breaks amino acids one by one from one end. That's called as exopeptidases. Okay. Now comes to the enzyme, major digestive enzymes of the stomach of the protein. In stomach, we have pepsin and renin. Okay. And in intestine, we have trypsin, chymotrypsin, elastase, carboxypeptidases, and aminopeptidases. Carboxypeptidases and aminopeptidases are exopeptidases. All other enzymes are endopeptidases. Okay, so this one, I hope you can understand now the answer for it. And more than that, if it goes more deeper, in which bond it breaks? Pepsin mainly breaks which type of peptide bond? Trypsin, which type of peptide bond? And it will be specified by some enzyme. So we are supposed to study that also. Okay, then another topic that should be covered is about the absorption of amino acids. After the digestion, for absorption, we need a cycle that takes place, gamma glutamyl cycle for the neutral amino acids. That word you should remember, makes a cycle. And this is all about digestion part. Inside a cell, intracellular protein digestion, we need another enzyme, cathepsin. That also you should remember. So some major points on digestion of proteins I was trying to tell. So here the answer is pepsin. Question number 10. All of the following are the products formed from the tyrosine except. So this comes to the amino acid metabolism now. As you know, melanin, epinephrine, dopamine, and of course thyroid hormones. Epinephrine, dopamine comes under catecholamines. They all are produced from the tyrosine. Serotonin is from the tryptophan. So the question was accept, please not. Just accept from the tyrosine. Now we'll go to the some option, this other questions related to this amino acid metabolism. First, you should know, see, about when we say about the amino acids, you should know the difference between essential and semi-essential, non-essential amino acid. Amino acid that cannot be synthesized in the body called as essential. We have 8 essential amino acids. We have 2 semi-essential amino acids. Try to study that. And you should know one thing, another one. So far, we were knowing 20 amino acids, but we have 2 more. 21st amino acid is called as cellulose-cysteine, and 22nd amino acid is called as spirolysine. So add to your knowledge, this question can come. And most of the amino acids can synthesize glucose. Only one amino acid is there purely ketogenic, that is leucine. That can be another question. If just ask you two amino acids, lysine and leucine, because lysine on breakdown can see what happens. One specific compound attached to lysine, then it breaks. So from there, we can synthesize glucose. But purely ketogenic is leucine. And derivatives of amino acid that are seen in protein. So not only amino acid, you can see some other derivatives of form. For one example, beta-alanine is one derivative of amino acid, different types of that. So that's what you should have an idea about that also. And another question can be amino acid with maximum buffering capacity. In our albumin, protein can act as a buffer. Which amino acid have the maximum buffering capacity? It is histidine. Because of its a group called as imidazole group have the maximum buffering capacity. So just try to say some important facts about the amino acids. Now we just go to our topic now. Glycine. From the that's the simplest amino acid, glycine. The major compound produced from glycine, one are creatine, creatine phosphate, creatinine. Say like that. Creatinine is an anhydrous form of creatine. Creatinine, the blood creatinine level help us to analyze kidney function. You know that. Creatine is stored in the muscle as creatine phosphate, a high energy compound, which help in the ATP synthesis. That reaction we call it as Lohmann's reaction. LOHMANN', Lohmann's reaction. Creatine phosphate help in synthesis of ATP through a reaction called as Lohmann's reaction. So these two, then glutathione, a tripeptide. When I say word tripeptide, that doesn't mean that it contains 3 peptide bond. It means that it contains 3 amino acids. Glutamic acid, glycine, gamma, glutamyl, glycinate, cysteine. So glutamic acids, glycine, cysteine. 3 amino acids combined together and form a compound called glutathione. It's an antioxidant. Just n number of functions. Okay, that's also a compound produced from glycine. And heme, the first step of heme synthesis, glycine plus succinyl-CoA giving ALA. So glycine is essential for heme synthesis. Then for the purine synthesis, purine mainly C4, C5, and N7 of purine. For that, we need glycine. So these are the major compounds produced from glycine. And of course, we know that it is also a neurotransmitter. It helps in its conjugation of bile salts. Or synthesis of bile salts we need glycine. Conjugation of bile acid. Okay, that also we need glycine. And for the detoxification of benzoic acid, we need glycine. That are some other functions of glycine. Phenylalanine and tyrosine. As you know, phenylalanine, with the help of by adding a hydroxyl group, it becomes tyrosine. So both helps in the melanin synthesis, thyroid hormone, as I said earlier, catecholamines. In catecholamine, we have norepinephrine, epinephrine, and dopamine. Norepinephrine, another word, noradrenaline. Noradrenaline, adrenaline, and dopamine. These 3 are together, we use the word catecholamines. And one more, on decarboxylation, produce tyramine. Okay. So these are some compounds from phenylalanine and tyrosine. And third major amino acid from which a lot of compounds are produced, we call as tryptophan. From tryptophan, of course, one vitamin, niacin. Tryptophan, amino acid from the amino acid tryptophan, a vitamin can be synthesized, niacin, and the neurotransmitter serotonin. Another hormone that maintains our daylight cycle, or diurnal rhythm is melatonin. These are three major compounds that are produced from the tryptophan. Of course, it can provide one carbon group is glucogenic. That's another important thing you should remember about tryptophan. And regarding tryptophan to niacin synthesis, I have 2 or 3 small facts there. Synthesis of vitamin niacin from tryptophan is inhibited by one other amino acid. It is leucine. Tryptophan and niacin synthesis, we need another vitamin. It is B6. That's a point to be noted. So I just try to say some facts regarding the compounds produced from different amino acids. I hope now you got the answer for the 10th question. The answer is an except, so serotonin. Okay. Going for next, another question related to metabolism of amino acids. Transamination is the first catabolic step of which amino acid given below. In general, transamination, transfer of amino group. Amino acid has 2 major groups, amino group and carboxy group. And the first step in the breakdown process of, or other word, catabolic process of amino acid is the removal of amino group. Okay, that's a common one. Okay, so that's about transamination. And what is the significance of transamination or importance of transamination? As we said here, it's the first catabolic step. Thereby, you can synthesize non-essential amino acids in this way. This process thereby can provide some precursor for gluconeogenesis. That means, for example, glutamic acid, when it undergo transamination, it produces alpha-ketoglutarate. It's a compound in TCA cycle. From that compound, you can synthesize glucose. So, transamination, another significance is it provides precursor for gluconeogenesis. And in most of the transamination, the compound that gets amino group is alpha-ketoglutarate. When you transfer the amino group from an amino acid, there should be somebody to accept it. The accepting one is alpha-ketoglutarate, mainly major acceptor is alpha-ketoglutarate, because it can undergo deamination at a faster rate. That's the point. Okay, now comes our point. Okay, in this transamination reaction, we need an enzyme called as transaminase. The major clinically important transaminase are AST, ALT. So here comes the question, what's the importance of these two? These two enzymes are elevated in hepatocellular damage. So enzymes through which hepatocellular damage can be identified is by analyzing the level of AST and ALT in the liver, in the blood. Okay, and coenzyme required is PLP. Now listen this one. This is a tricky one. Amino acid that cannot undergo transamination are proline, lysine, threonine. That means what? These amino acid cannot undergo transamination. So for these 3 amino acids, transamination cannot be the first catabolic step. Now we can check the question now. Transamination is the first catabolic step of which amino acid given below. Proline, threonine, lysine cannot undergo transamination. So the options are ruled out. Then we have only one, arginine. Other words, all other amino acids out of 22, all of the other amino acids can undergo. Or the main amino acids, 20 amino acids, all other 17 can undergo transamination. Or that's the first catabolic step. That's what. So here the question indirectly asked was exceptions in transamination. Which all amino acids cannot undergo transamination? That was the question asked. Okay. Then after transamination, we have a process, one more thing, trans-deamination. Just to know what is trans-deamination. Transamination, when coupled with oxidative deamination, is called as trans-deamination. That also you should know. That happens in liver. Okay, we'll go to the next 12th question. synthesis of 1 mole of urea requires how many moles of ATP? In other words, it's about the energetics of urea cycle. Synthesis of urea takes place through urea cycle, you know it. Okay, so first of all, what is urea cycle? Just now we said synthesis of urea. Where it takes place? In the liver. Why it takes place? Because during the amino acid catabolism, there's a transamination, then deamination. So this amino group is removed as ammonia. Ammonia is neurotoxic. So this neurotoxic ammonia is converted to less toxic urea. That's the point. So site is liver. Use is to detoxify. Urea cycle is to detoxify ammonia. That's the point. And you know, the source of ammonia is not only amino acid. Breakdown of purine, then the amino group in the purine is removed as ammonia. Breakdown of pyrimidines, that's another source of ammonia. Then the action of bacteria on the food particles in the intestinal bacteria's action, that's another source of ammonia. So that all can be the part of a question. Then how the ammonia is transported? As what? Mainly from the brain is transported. Many other tissues have transport as glutamine. And from muscle alone, the ammonia is transported as alanine. That's another 2 facts you should know. When the question is energetics, actually the question is asking on the relationship with TCS cycle. Okay. I'll come to that word now. Clinical importance of urea. What's the clinical importance of urea? Urea's normal level is around 20 to 40 milligrams per deciliter. This will get elevated in kidney dysfunction. So urea is also another biochemical parameter that helps to analyze kidney function. So 2 major, the routinely used parameters to analyze the kidney functions are urea and creatinine. That's a clinical importance of urea. Now, this is about the urea cycle. In the urea cycle, see, there are 2 steps. One step, 2 ATPs are utilized, and another step, 1 ATP is converted to AMP. So this is equivalent to conversion of 2 molecules of ATP into 2 molecules of ADP. Okay. So here also 2 ATPs are utilized. Here also 2 are utilized. So 2 plus 2, 4 are utilized. So we may think that 4 ATPs are needed for urea synthesis, of course. But the matter is the fumarate that is produced in this urea cycle, released out, that enter into the TCS cycle. Enter the TCS cycle, and in the TCS cycle, this fumarate will be able to produce 1 NADH. 1 NADH can generate 2.5 ATPs. Okay, so through urea cycle, 4 ATPs are utilized. Because of its relation with TCS cycle, it is able to produce 2.5 ATPs. So the net utilization is 1.5 ATP. So your answer is 1.5 ATP. For synthesis of every molecules of ammonia, we utilize 1.5 ATP. That's the answer for that question. So the examiner is trying to evaluate your understanding about the urea cycle's relation with TCS cycle. I try to almost all the important questions about urea cycle I try to be able to explain. Going for the next one now. Which of the following lipoprotein transport cholesterol from liver to peripheral tissues? And same way, another question. Which lipoprotein is called as good cholesterol? First of all, the word lipoprotein is a combination of lipids and protein. What's the use of it? Help in the transport of lipids from one tissue to another, because lipids are insoluble in the water. Most of them are insoluble in water. So it should be transported with the help of somebody else. Proteins will help in its transport. And mainly the major lipoprotein present in our blood are chylomicron, VLDL, or another word very low density lipoprotein, low density lipoprotein, and high density lipoprotein. So 4 we have. Out of which, understand the functions of each. Lipoprotein in the chylomicron transport the dietary lipids, the lipid what we have, to the diet. So it will be transported from intestine towards tissues. Okay. And then other one, next is VLDL. VLDL transports endogenous triglyceride. That means what? The triglyceride that are produced in the liver, from liver towards tissue. LDL helps the transport of cholesterol from liver to tissues, and HDL transport cholesterol from tissues to liver. See, it transports, the first 3 were transporting towards tissue, and this alone from tissues to liver, just opposite side, reverse. So HDL transport is also called as reverse transport of cholesterol. And the one important point to be noted is cholesterol is inside in every cell. For its excretion from the body, or to prevent the accumulation of the cholesterol from the body, it should be brought towards liver. From the liver, the cholesterol is mainly excreted as either 2 compound cholestanol, coprastanol, or and as bile salts. Okay, so it should come to liver for the excretion through the bile. So any compound, any molecule that brings towards liver, that brings cholesterol towards liver, is helping in its excretion, thereby prevents the chances of atherosclerosis. So that's called as a good cholesterol. So because of this property, HDL, since it transports cholesterol from tissues liver, it's called as good cholesterol. And LDL is transporting cholesterol from liver to tissue, so it's causing accumulation of cholesterol, so it's called as bad cholesterol. This thing you should know. And regarding the transport of cholesterol from tissues to liver, we require PUFA. What is PUFA? Polyunsaturated fatty acid, or essential fatty acids comes under that. That points also you should not. So here in both questions, in 2 ways it is asked, and the answer is HDL, high density lipoprotein. Okay. Which thing is false about trans fatty acid? First of all, what is fatty acid? Fatty acid means carboxylic acid, aliphatic compound containing a carboxyl group. That's fatty acid. We will say some major divisions. Essential fatty acid are mainly linoleic acid, linolenic. Like our essential amino acids, we have essential fatty acid also that cannot be synthesized but should be supplied through the diet. They are linoleic and linolenic acid. Okay, they comes under PUFA only. That's what I was trying to say. They help in the transport of cholesterol from tissues to liver in the HDL metabolisms. Based on the number of, so one division based on the nutritional importance, essential and non-essential. Based on the number of double bonds or absence of presence of double bonds, saturated fatty acid, monounsaturated fatty acid, and polyunsaturated fatty acid we have. Then another thing, any fatty acid having a double bond will be in it can exist in 2 forms in nature, cis form and trans form. And all the naturally occurring fatty acid will be in the cis form. See, trans form we always see in the highly processed food and bakery items. Okay. This trans fat is very not good for the body. It can cause endothelial dysfunction. It can cause insulin resistance. So thereby it can cause diabetes. It can cause adiposity. Okay. And here, This is what most of the trans fatty acids are produced during hydrogenation of unsaturated fatty acid. So when you understood this fact, I hope that your answer is ready now. So hydrogenation of fatty acid. Hydrogenation increases the amount of trans fatty acid in the nature. That's the answer. 16th question. Heme in the hemoglobin. About it. What's all about? You know that heme is the non-protein part of hemoglobin, or other word, prosthetic group. This heme and globin chain together form the hemoglobin. We have 4 globin chain. In a mature hemoglobin, adult hemoglobin, HbA, we have 2 alpha and 2 beta. We have one, that's around 95 to 97% is HbA1 or HbA in general. We have HbA2 also, alpha 2, delta 2. And in fetal hemoglobin is alpha 2, gamma 2. Some 1% is there in our body. So anyway, we have 2 alpha chain. Normally we have HbA1 contain 2 alpha chain and 2 beta chain. Each globin chain will be attaching to one heme. That point you should know. So 2 types of globin chain we know, 2 alpha, 2 beta. Alpha chain contain 141 amino acid. 7 helical segments are there. The 2 major secondary structure of proteins are one helical, other one is beta pleated sheets. 2 major secondary structure. In that, alpha chain contains 7 helical segments, and we just named them as A, B, C, D, E, F, G. And beta chain contains 8 helical segments. It contains 147 amino acid, A2H. And understand that heme is present between E and F helices. In the heme, at the center, you have an iron atom. Iron atom attached to protoporphyrin ring. Iron Fe2 plus have 6 valencies. 4 are attached to porphyrin. 5th will be attached to, we call it as a histidine near to it. So the word proximal histidine at the F8. That's common. And 6th one, in the oxyhemoglobin, the 6th valency of the F ferrous ions is attached to oxygen atom. Oxygen atom then also attached to distal histidine of E7. So the heme is present between E and F helices. Now, I hope almost all the answers are here. Hemoglobin with protoporphyrin 9. That should be the answer now. Okay, so I hope the answer is okay now. Heme non-protein part attached to every globin chain in between the E and F helices through histidine atom. Histidine is basic, so it's polar only. Histidine is polar, is the basic amino acid. So C is also, now we can rule out that. Answer is protoporphyrin 9 with iron atoms. Okay. Which of the following is false regarding sickle cell hemoglobin, HbS? HbS. You know that sickle shape hemoglobin, where the beta chain globin chain at the 6th position, if glutamic acid is replaced by valine, that's called as sickle cell hemoglobin. And it forms a sickle shape only in the deoxy conditions. RBC form a sickle shape, not the hemoglobin. And there comes a word, a common term, hemoglobinopathy. Any diseases related to hemoglobin you call it as hemoglobinopathies. Abnormality in primary sequence is that means what is primary? So structure of protein, mainly we have primary structures, secondary, tertiary, and quaternary structures. Primary structure means the number of amino acids present in a protein and the sequence in which it is arranged. So we'll say number and sequence of amino acids in a protein is called as primary structure. Now here what happens in sickle cell hemoglobin? In that sequence, in the number is same, but the sequence there's a small change. At the beta chain, 6th position, instead of glutamic acid, valine comes. So abnormality in the primary sequence. So that's one type of hemoglobinopathy. Then we also call it as qualitative hemoglobinopathy. Another one, quantitative hemoglobinopathy, where chains what are synthesized are normal, but the synthesis rate might be insufficient. The synthesis of beta chain synthesis might not be equal to the rate of synthesis of beta chain might, if it is not equal to that of alpha, then we call it as beta thalassemia. And sometimes alpha chain synthesis, the rate will be less than that of beta. We call it as alpha thalassemia. So we generally quantitative hemoglobinopathy. Thalassemia is quantitative hemoglobinopathy. Please note. Okay. Now come back to our question at BS. In the beta chain, 6th position, there's a conversion of glutamic acid to valine. GAA is replaced by GUA. That's a genetic code difference in that. What is codon? What are codons? What is GAA? It's a trinucleotide sequence that code for one amino acid is called a genetic code. Here GAA that is able to code for a glutamic acid. Now in our DNA, because of some mutations, GAA becomes GUA. Naturally, then what will happen? Then it is replaced by valine. Okay, glutamic acid replaced by valine. Now here what happened? Because of the change in the globin chain, beta chain, in an RBC, in deoxy condition, all this hemoglobin will aggregate together, clump together. That leads to sickling of RBC. So now question comes, why RBC sickles? And when RBC form a sickle shape in only in deoxy hemoglobin, deoxy state or deoxygenated state. Why it happens? Because the sickle cell hemoglobins all will aggregate or clump together. Both are important. How many hemoglobins are in the RBC? The answer is not 3 or 4. 75% of the dry weight of RBC is hemoglobin. So we contain n number of hemoglobin in one RBC. These hemoglobins will combine together, aggregate together in only one condition, deoxy conditions. Then it forms a sickle shape. So HBS forms only during, sickle cell RBC formation happens only during deoxygenated state. That's also you should remember, leading to sickling of RBC cells. So aggregation of our hemoglobin leads to sickling. Okay. Now next question, tetany is manifested in. As you know, tetany is regarding calcium. What's the normal serum calcium level? 9 to 11 milligram per deciliter. And then here comes our answer, some important points about our answers. The 9 to 11 calcium level, that, okay, take it as 10. That exists in 3 forms. 50% as ionized form, just as calcium ion, Ca2 plus ions. So that is metabolically active. Around 4% is attached to albumin. That's called as protein bound form, 4 milligram, around 40%. Another one, 10 milligram, another 1 milligram, 5 plus 4 plus 1 in the complex form with carbonate, citrate or phosphate. Okay. Now, please understand that out of 10, only 5 is metabolically active, this ionized form. Now, what are the factors that maintain calcium level? Here we have hormonal factors and non-hormonal factors. Hormonal factors are 3 hormones: parathyroid hormone, calcitonin, and active form of vitamin D that also act as a hormones. These 3 are the ones which mainly regulate the blood calcium level or serum calcium level. And the non-hormonal factors, please note, acidosis, alkalosis, serum phosphorus level, age, children, till more, serum protein. Now our point comes to, now we are going to settle down to acidosis and alkalosis. In these conditions, proteins will be having extra some charges. Acidosis will be having plus charges. Alkalosis will be, the protein will be having negative charges. We earlier said that 40% of the calcium is attached to the protein. Now, in alkalosis, what happens? Number of negative charges will be more. So more calcium will be getting attached to it. The ionized calcium, when the pH become alkaline, the protein will be having more negative charges, so more Ca2+ will go and bind. So what happened during alkalosis? That's the point. During alkalosis, the level of metabolically active calcium ions will be less because most of them are getting attached to serum proteins. So the metabolically active calcium will be less in alkalosis. When the hypocalcemia occurs, that leads to tetany. So alkalosis favors the binding of ionized calcium to protein, decrease the level of ionized calcium. So that's the point. So you should know that tetany occurs when the serum calcium level decreases. What's the normal level of calcium? In which all form it exists? And what are the factors it regulates? If you know these points, then only you'll be able to answer this question. Or other word, hypercalcemia can occur when? In all acidosis condition. Hypocalcemia can occur in alkalosis condition. That's another point to be remembered. Hope you understood that. Okay. Next question. Choose the dietary source of vitamin C from the following. No doubt, vitamin C4 from citrus fruits. So the main source of vitamin C is, as you know, amla, our nellika, then guava, perakia, then citrus fruits. So source of vitamin C is vitamin C is also called as ascorbic acid. And then functions, most important one, vitamin C for collagen. Understand like that collagen cross-linking. That's the main reason for our most of the functions. And it also helps in iron absorption, conversion of methemoglobin to hemoglobin. So for the conversion of methemoglobin to hemoglobin, you need 3 compounds. Please note, just now we said earlier NADPH, now vitamin C, and third is glutathione is essential for it. So for the conversion of methemoglobin to hemoglobin, we need 3 compounds. What they are? NADPH, vitamin C or ascorbic acid, and another one, glutathione. Okay, that's a antioxidant property. Now listen, deficiency manifestations. See, our blood vessel contain collagen. Collagen cross-linking and maturation requires vitamin C. So whenever there's vitamin C deficiency, the blood capillaries become fragile. A small tap is enough. It causes the breakage of the capillaries. So subcutaneous hemorrhage may occur in vitamin C deficiency. And the same way, we have a spongy gums. Collagen is not mature. So bleeding gums, spongy gums. That's another reason for the vitamin C, another manifestations of vitamin C deficiency. Then the bonds are fragile because bonds also contain collagen. So scorbutic or weak bonds is another feature of vitamin C deficiency. Another feature is it can have anemia. Why? Because iron is not getting absorbed, so heme formation is reduced. One thing. Then what all the iron are there? What all hemoglobins are there? Or what are RBCs? Because of hemorrhage, we lost RBCs, so hemoglobin is we are losing that because of hemorrhage. So because of this reason, there will be anemia too. Hypochromic microcytic anemia, because our hemoglobin is less, hemoglobin gives the color, less color. Vitamin C deficiency leads to the decreased level of hemoglobin. So hypochromic, when the hemoglobin level is less, RBC size reduces, so hypochromic microcytic anemia. That's about the question on vitamin C. Important question, please note then. Another one. Okay, deficiency of which vitamin has been associated with enamel defects and increased risk of dental caries? So one by one, we'll say vitamin D. Active form is calcithiol. Calcithiol acts like a hormone. Just now I said it maintains blood calcium level. three hormones that maintain blood calcium, just I was telling now, PTH, calcitonin, and calcitriol, by acting on three sites, on the bone, on the T, on bone, in the intestine for the absorption, reabsorption in the kidney. So in this way, calcitriol try to maintain the blood calcium level. And it also plays a key role in bone and teeth mineralization. And when the levels are unregulated, it can lead to racketing. That means breakable tooth, which is the defective one or hypomineralized organ, highly susceptible to fracture and decay. So this another property, another function related to vitamin D, not only in bone mineralization, but also helps in the teeth mineralization. So vitamin D deficiency is the one that leads to enamel defect and the increased risk of dental caries. Okay, we'll go for other things also. What is the use of vitamin A? Mainly we say it exists as retinoids, like calcitriol, we said retinol, retinal, retinoic acid. We know it helps in vision, not only in vision, it also helps in cellular differentiation, epithelial maintenance, decrease keratin synthesis. This all should remember. Folic acid helps in one carbon metabolism, thereby cell division replication. That's another use of folic acid, is the use of folic acid. Simpler biochemically, we say folic acid helps in one carbon metabolism. Thereby, more generally, we can say it helps in purine pyrimidine synthesis. Purine pyrimidine for DNA RNA synthesis, or other word nucleic acid synthesis. Thereby, cell division and replication. Okay, that's what about the folic acid. Vitamin C we have already said, vitamin D also we said. So here the answer is vitamin D. Option D is the answer. All vitamins play a role in citric acid cycle except, that's another question. Here, citric acid cycle and in glycolysis, glucose converted to pyruvate. Pyruvate to acetyl-CoA step. That step is very equivalent to that of, biochemically equivalent to that of conversion reaction, alpha ketoglutarate to succinyl-CoA. In all these steps, we need some vitamins, coenzymes, active form of vitamin. Thymine, active form of TPP, riboflavin, FAD, FMN, niacin, NAD, NADP, pyridoxine, PLP, pantothenic acid, coenzyme, folic acid, THF, biotin for another vitamin. We have biotin itself, cobalamin, B12. It exists as cyanocobalamin. It has a methylcobalamin and adenosine. So these vitamins act right. The vitamin B complexes, they act as they exist in active forms. We call it as coenzyme, which help in the action of enzymes or helps enzyme for its action. And in alpha ketoglutarate dehydrogenase, a step in TC cycle, and conversion of pyruvate to acetyl-CoA. The acetyl-CoA is the compound that enters into TC cycle. So in these two reactions, we need 5 coenzymes and 3 enzymes. The 5 coenzymes involved are thymine, riboflavin, niacin, and pantothenic acid, the major enzyme, major vitamins. And we say we have one more one, lipoic acid, also considered as early vitamin earlier. So these 5 compounds we need for that enzyme. So the major vitamin that play a role in citric acid cycles are riboflavin, pantothenic acid, vitamin, and niacin. Ascorbic acid is not involved in citric acid cycle. That's the answer to be noted. Okay. Next question. One of the following tests is not useful to assess kidney function. Already I was telling about these two, urea and creatinine. And one more component test is, I'll say one by one now. Renal function test, mainly we use urea, another one creatinine. And urea, the problem is this value may change with our protein diet. So there are some extra renal factors that can vary the urea level. So urea is not a perfect one, but urea can be utilized. Second one is creatinine. But one important thing about creatinine is that, one disadvantage is that In mild renal impairment, that means the starting stage of renal impairment, there will be more, there's not only filtration, tubular secretion is there. Tubular secretion increases, so the creatinine value will not vary in the starting stages of renal impairment. That also can be used as a parameter for renal function. These two, both urea creatinine, but it has its own disadvantage. That's what I was trying to say. Third important compound is cystatin C. Just please note that it's a non-glycosylated protein. That means a protein without any glucose, without any carbohydrate. 0.8 to 1.2 milligram per liter is the value. And it is produced at a constant rate by every cell, freely filtered, completely reabsorbed and degraded in tubules. So it is getting totally filtered out by the kidney. So if the kidney is functioning well, it will filter out. It will be complete and then degraded by tubules also. So that is now a better marker of renal function than urea creatinine. Because the cystatin C is sensitive in that area where the mild renal impairment, we call it as creatinine blind area. In that area also, cystatin C is sensitive. So these three can be used to assess kidney function, out of which cystatin C is the best. Now the other one, plasma creatinine, is an hydrous form of creatinine. It is not at all used for kidney function. That's what you should know. Now one more I would like to add up now to know the renal function test. Nowadays we have another one also, urinary neutrophil gelatinase associated lipocalin. We call it as NGAL, UNGAL. Early detection of any acute kidney injury, we can use that also. Some major factors. Okay, that's all about the kidney function now. Another one, sigma subunit of prokaryotic RNA polymerase. This is a question about the transcription. For transcription, we need a major enzyme called as RNA polymerase enzyme. That you should know. Then RNA polymerase have 5 units. Okay, I'll put it this way: 2 alpha, 1 beta, beta dash, omega. These 5 units together form the core enzyme and help in the elongation part of our transcription of our RNA synthesis. It has extra one called sigma subunit. Together we use the word holo. The core enzyme plus sigma we call as holo enzyme. Holo enzyme is essential for the starting of transcription. So sigma subunit is the one which is able to understand the starting point. Okay, through binding to a site called as promoter site. So promoter site gives a signal about your starting point. Sigma subunit binds to the promoter site. So in that way, that's important for the initiation process. Elongation process is done by core enzyme. Okay, that's the thing you should remember. And termination, it's not given here. Initiation by sigma, elongation by holo enzyme, and termination by another protein called as raw protein. Like sigma, we have another protein, raw protein. Please note that also. Okay, this is about, and here, please note the beta in that 5 holo enzyme. Beta is helping in the catalyzation process. But that means the addition of nucleotides to form an RNA. That is catalyzed by beta subunit. Okay, and of course, some inhibitors of this one transcription can be asked. Same way, inhibitors of replication, translation can be asked. Please note the things. Okay, next question. Leishman syndrome is due to, first of all, what is all about? Purine synthesis in the body. There are 2 types of purine synthesis. One is we call it as de novo synthesis. Another one, salvage pathway. De novo means synthesizing a new one using all the components. Like building a wall using bricks, like that, one by one, adding up each atom, each molecule, and making a purine. Salvage pathway is a recycling process. That means already produced nucleotides, breaks, and make it excreted out. It is prevented from excretion and is recycled. For that, we need 2 method enzyme, APRTase, adenine phosphoribosyl transferase, and HGPRTase. H stands for hyperstanding. Hyperstanding guanine phosphoribosyl transferase. This is involved in salivary pathway. And the deficiency of HGPRTase deficiency leads to a condition called as Lesch-Nyhan syndrome. And here, the most important signal or clinical manifestation is self-mutilation. That patient will be biting and chewing his own lips, his own fingers, and hurting himself. And hyperuricemia, increase of uric acid. So the gout may occur. And here we have an orange diaper disease. Because uric acid crystals is orange color, the diaper will be orange in color. We have alkaptonuria where black color urine, black diaper is orange diaper disease. Please note. Our last question. The most abundant glycoprotein in the base membrane. Hope we know about that. Extracellular matrix is about the interstitial matrix present between the cells, or it's about the base membrane on which the epithelium and the endothelium rest. These all we use the word extracellular matrix. It contains mainly type 4 collagen. That's a major structural component. Contain mainly 3 groups of protein. One is our fibrous protein. This is mainly our collagen. Then water hydrated gel. That is nothing but our heparan sulfate provides a charge barrier to limit the passage of molecules. We have something for the glycoprotein for the attachment of the cells. That is laminin. Provides a mechanism for the attachment of epithelial cells to the collagen. Also, we have nidogen or endactin. The function remains, we don't know exactly, but we say it along with the laminin helps in the process. So if I ask you the major glycoprotein, that's laminin. It's the major glycoprotein present in the extracellular matrix or mainly in the base membrane. And heparan sulfate is the major mucopolysaccharide. Mucopolysaccharide in that, mainly in the base membrane. So based on the questions, the answer varies. Since here the question is glycoprotein, is laminin. Okay, that helps in the attachment of epithelial cells. So I hope you understood now. You're able to understand these things. These are some major questions that has already been in our asked so far. So all the best for your future for the exams. Hope you enjoyed this class. You're able to answer. Now you'll be able to, I tried to not only answer the questions, but I've tried to say some other questions related to that questions, to that topics itself. Hope you enjoyed it. And thank you for the patient listening. Thank you.